Hi, welcome everybody. Well, people are trickling in and are still enjoying their, some of their lunch, so they'll be here shortly. But for the sake of time, we have to get started because there's also a session right behind us. My name is Serge Kremers. I'm chairing this session. I'm a professor at Columbia University in New York City. I'm also the editor-in-chief of British Journal of Clinical Pharmacology. It is my honor to introduce Dr. Sofia, Michael Sofia. He is the co-founder and the chief scientific officer of Arbutus Biopharma, where he established the programs in hepatitis B virus cure and coronavirus cure therapeutics. During his career, he has introduced over 20 drugs into clinical development for both infectious disease and inflammatory diseases. He is responsible for the discovery and early development of sofosbuvir, which became the backbone of many hepatitis C virus curative therapies. Mike has held research and research management positions at Gilead, Pharmasset, Bristol-Myers Squibb, Transcell Technologies, and Eli Lilly. He holds a BA in chemistry from Cornell University, a Ph.D. from University of Illinois Urbana-Champaign, under Prof. Katzenellenbogen, and of course, most importantly, he was the NIH postdoc fellow at Columbia University in the labs of Professor Gilbert Stork. Mike sits on the scientific advisory board of Foresite Labs and is a trustee of St. Joseph's University in Philadelphia. I just happen to know they have a great basketball team. And Mike has authored over 120 publications, 4 to 15 book chapters, 54 US patented, and has edited special journal issues on infectious disease. He's received numerous awards and is really a superstar. He received the Lasker Award in 2016, and most recently he received here in Edinburgh the Cameron Prize for Therapeutics from the University of Edinburgh. In 2016, he was inducted into the American Chemical Society Medicinal Chemistry Hall of Fame, and he's a fellow of the Royal Society of Chemistry. And he's going to talk about hepatitis B and C virus cures, hopefully. So thank you, Mike.
Great, thank you for this introduction. And thank you to the organizers for the invitation to come here and tell you a story that sort of began probably about 20 years ago, but now sort of continues to go on in a new phase. Okay, so viruses, to give you a little background, are not living, right? They must be infectious to endure in nature, and they're sort of obligate intracellular parasites. They cannot make their own energy or protein, so really rely on the human or host machinery to do what it needs to do to survive. Most interesting, they possess many ways of subverting the host's immune response as part of their existence. So there are a number of chronic viral infections shown here. And these chronic viral infections really rely significantly on either virus-host immune control or some type of latency where there's a very low replication going on in some part of the body, such that a certain activation event will ultimately increase that replication and have the virus continue to survive. Now, we're going to talk about viral hepatitis here. And so viral hepatitis really is an inflammation of the liver. Its symptoms are abnormal pain, nausea, fatigue, et cetera. Major causes is that, in fact, 50% of the causes of hepatitis really are viral in origin. Turns out there are 5 different types of hepatitis viruses, A, B, C, D, and E, but there are only really 2 of these viruses, hepatitis B and hepatitis C, that actually lead to chronic disease. Hepatitis delta, or D, is dependent solely on hepatitis B to be to exist because it relies on a certain protein from hepatitis B to actually enable its life cycle. Hepatitis A and hepatitis E are self-limiting, caused by either ingesting contaminated foods or water. But if you look at hepatitis B and C, which you see that they are a major public health problem globally. In fact, every third person on the planet has been infected with hepatitis B at some time in their life. Some resolve the infection, sort of basically an acute infection. Others go on to have chronic disease. In fact, there are 500 million people that are currently chronically infected with hepatitis B and hepatitis C globally. 1.3 million people die. 80 to 90% of all liver transplants globally are associated with hepatitis B and hepatitis C infection. And so the majority of chronically infected are undiagnosed, and therefore many people go many years without really knowing whether they have this chronically infected disease. Now, if you look at the mortality rate of individuals infected with various infectious diseases, what you see is that over time, begin to see the reduction in mortality in a number of major viral infectious diseases, including malaria, HIV, and tuberculosis. But the only viral infection that continues to grow turns out to be viral hepatitis, and in fact is projected to grow exponentially into the future. And a lot of this has to do with, in fact, that viral hepatitis, the opioid epidemic, particularly in the United States, is causing tremendous increase in both hepatitis B and hepatitis C viral infections. So about 10 years ago now, actually in 2013, the first interferon-free cure for hepatitis C was identified. That drug, known as sofosbuvir, commercially as a commercial name called Sovaldi, ultimately led to the ability to cure patients in 8 to 12 weeks of therapy, with 8 to 12 weeks of oral therapy, and leading to very high cure rates. And in fact, today, if you look at the number of drugs now currently approved for curing hepatitis C, in fact, 6 of the 10 drugs require sofosbuvir as part of the combination, so it is the backbone. In fact, 95% cure rates, or greater than 95% cure rates for these DAA, or direct-acting viral combination. And again, one pill once a day for 8 to 12 weeks. And the outcome is essentially 65% reduction in liver-related deaths in the US, and the direct-acting virals for HCV cures have been associated with a 79% reduction in hepatocellular carcinoma risk, basically 50% reduction in HCC cases in the United States. So what you see now, the WHO has now, several years ago, initiated an effort to ultimately eliminate or eradicate hepatitis C globally based on the use of these direct-acting viral agents. And today, the two major direct-acting viral agents that are being used for cure are Epclusa, which is sofosbuvir-velpatasvir combination, and Mavorette, which is is a sort of a protease inhibitor plus an NS5A inhibitor. But when you look at hepatitis B, it's a different story. So if you look at hepatitis C is a virus. It's an RNA virus. It replicates only in the cytoplasm of the liver cell. It does not have any integrated form of its genetic material and has no reservoir. So in essence, it was a problem that was, although it took many years to solve, was more easily solved than hepatitis B. And the problem with hepatitis B, in fact, is that it has a viral reservoir, so it has this thing called covalently closed circular DNA, which resides in the nucleus of the liver cell. It's a very long-lived, very stable form of DNA. And small segments of the viral DNA also integrate into the human genome and ultimately produce from that, not necessarily replicative virus, but does produce important viral proteins. If you look at the viral genome, it's an interesting viral genome. It's a number of overlapping reading frames. which will be important actually in some of the discussions on how you can produce a therapy for this. Sorry, technical difficulty. So let's look at the virus life cycle and understand what the complexities is associated with the hepatitis B virus. So first, the virus life cycle is very complex. The virus actually enters the cell through the NTCP, or sodium taurate co-transporter polypeptide. It enters into the cell, loses viral coat, and then deposits this thing called relaxed circular DNA into the nucleus of the liver cell. Now, the virus then takes advantage of all the DNA processing enzymes or DNA repair enzymes that form this thing called cccDNA. And from there, all the downstream viral proteins and viral genetic material is formed. You form a thing called pregenomic RNA that then gets encapsulated into a viral coat, a viral capsid structure. And that pregenomic RNA, including the viral polymerase, then forms what is known as the relaxed circular DNA. The relaxed circular DNA can achieve a viral coat and then be extruded from the cell, or it can actually be recycled back into the nucleus to replenish this pool of cccDNA. So you have continual replenishment of this genetic material that's the sort of reservoir of viral genetic information. You also, one of the other things that produces from cccDNA is this protein called S-antigen. So S-antigen coats what are known as subviral particles as well as the virion itself. And what's interesting about S-antigen, if you look at the high replication rate of HBV, HBV is produced about, in a chronically infected cell, you produce about 10 to the 12th new virions per day. Well, you turn out that you produce 10,000 times higher quantities of these subviral particles coated with S-antigen. And it is believed that the S-antigen plays a significant role in the way this virus controls the host immune response by a process called immune exhaustion. So what you have is a very high rate of replication. You have this pool of transcriptionally active cccDNA that's very stable. You have a large production of the immune tolerizing S-antigen, and you have this HBV-specific T cell and B cell silencing that is a result of this production of S-antigen. So, how are you going to address a cure for hepatitis B? So what we do know is, well, today, there are therapies for hepatitis B. They're maintenance therapies. So you can have a nucleoside, tenofovir and entecavir, tenofovir alafenamide, that are all lifelong therapies that suppress viral replication. They don't cure the patient, they just suppress viral replication and do have a positive effect on the reduction of your incidence or chance of getting hepatocellular carcinoma. But how do you cure this? How do you cure it with a therapy that is a finite duration therapy at a high cure rate? So the proposal that we've been working on for a number of years now is shown here. And we believe you have to do three key things. One is you need to completely suppress viral replication and reduce the pool of cccDNA. The second is you need to reduce this viral antigen pool dramatically, hopefully enabling the host immune system now to re-engage, and then possibly bring in an immune activator to ultimately reactivate the quiescent T cells and B cells. This ultimately, we believe, will lead to a functional cure. Now, a functional cure is not a sterilizing cure because the cccDNA today, even today, there is no known way of eradicating or eliminating cccDNA from the cell. So basically, a functional cure is undetectable HBV DNA, undetectable S-antigen levels with or without the production of S antibodies after cessation of all therapy after 6 months. So that's what our objective is today. So let's talk about the viral replication process. So as I mentioned today, nucleosides are able to inhibit the replication process that takes the pregenomic RNA and makes cccDNA. There are several drugs, tenofovir and entecavir, that are able to inhibit at this part of the pathway. But what we do know is that it's leaky, right? So you still get production of new virions coming through. You still get replenishment of that pool of cccDNA. So the hypothesis was, can you add a novel mechanism of action agent into this mix, this combination, to ultimately shut down this process completely? And that was proposed, that proposal led to this series of molecules known as capsid assembly modulators. And so capsid assembly modulators will basically inhibit the encapsulation of pregenomic RNA as another mechanism to shut down replication, as well as the ability to inhibit the uncoding process in the release of relaxed circular DNA into the nucleus of the liver cell, producing new cccDNA. So if you look at the capsid structure, it's actually 120 dimeric core proteins associated with it, and they are critical for maintaining the infectious virion production and maintenance of nuclear cccDNA. So early on, there were a series of molecules that were identified known as capsid assembly modulators, and they're actually in two different classes. One is known as the CAM-A or CAM-aberrant or CAM-E or CAM-empty. And what does that mean? So it turns out that these molecules bind a core protein, and they either form aberrant polymers of core proteins so that you don't have that capsid structure, or actually you form empty capsids. So there are these two different classes of molecules that turns out ultimately achieve the same end, but by slightly different approaches. So we've been interested in the CAM-E strategy for quite a while, and actually introduced into the clinical development this molecule, AB423, a modestly potent capsid inhibitor, first generation agent as we called it. And it went into clinical development. Phase 1 clinical data showed that the PK was not optimal. We had BID dosing. It had multiple metabolites associated with it. So we decided to abandon this and go to a second generation known as AB506. This molecule was significantly more potent than the first generation agent. And this molecule also went into clinical development. It was a once-a-day dosing frequency, had very good PK, showed very nice viral load decline in HBV DNA and HBV RNA levels, as one would hope and expect. But ultimately, we identified a ALT elevation that occurred on a day 28 study in chronically infected patients. You saw ALT elevations in the grade 2 and grade 3 ALT elevations at about day 20. Now, what you have to know about HBV patients is that they always have ALT elevations that occur during their chronically infected state. And so one doesn't know whether an ALT elevation is a bad ALT associated with the drug or whether it's associated with reactivation of a host immune response, or what they call a positive ALT flare. So we took this drug back into the phase 1 study to answer that question, a 28-day phase 1 study, and in fact it showed an ALT elevation at day 20 in healthy volunteers, indicating there was some kind of a toxicity associated with this chemical series. In addition, this chemical series also had a resistance pop up. So there was a patient that was a non-responder, an I105T variant, that indicated that this molecule had some liabilities associated with resistance. So we went on to generate a third compound known as AB836 that we wanted to have improved potency, in vivo efficacy, PK, and obviously safety profile. Now, this was a DILI, we believe, associated ALT tox, ALT elevation, liver tox, but we didn't know really what caused it. So what we wanted to do is then develop a completely different chemical series And that's why this chemical structure, you see predominantly the whole right-hand portion of this molecule is very different from AB506, hoping that we would circumvent that DILI toxicity. So if you look at these, actually, this is a co-crystal structures of overlapping co-crystal structures of showing these very different this is the sort of the CAM-A, this is the first, second, and third generation. And what's very interesting about this is that they all bind to the same binding site on core protein at the dimer-dimer interface. Even though as well as the sort of CAM-A or CAM-E, they all have very different outcomes. One forms empty polymers, sort of empty capsids, and the other one forms linear aberrant polymers. So if you look at the crystal structure shown here, this shows the binding of this third generation agent that I talked to you about, AB836. And what you see is there's an important hydrophobic binding clef in here requiring this substituted aromatic and key hydrogen binding interactions shown here. And then this other moiety on the far right portion that I showed you in the previous slide binds in a sandwich portion between these two residues. Now, what that does, what this additional binding characteristic does, it enhances its potency, but also gives it a very dramatically improved resistance profile that I'll show you later. So the molecule AB836 was shown to be quite potent in low nanomolar active agent against inhibiting viral replication, the production of rcDNA, as well as the second mechanism, right, inhibiting the replenishment of cccDNA, and the S antigen, production of S antigen is a biomarker for that outcome. The molecule was shown to be pan-genotypic. It's been potent activity against primary and secondary mechanism, as I just showed, and only about a 2.3-fold decrease in potency against 40% serum in vitro. It was very selective for HBV. We tested it against probably 10 or 15 other RNA or DNA viruses and saw essentially no activity. And what you see with these on a gel is that you can see that 836, so this is entecavir, this is a nucleoside. GLS4 turns out to be a CAM-A type inhibitor. But what you see is that you have no encapsulated pre-genomic RNA here. You also see core DNA is not there. So this is the rcDNA is not there. But you still have core protein. So you still have the capsid structure intact, where in the CAM-A, you see that capsid, the core protein is no longer there. And all these other proteins still exist in the entecavir model. So showing the mechanism as we had originally hoped. Now this molecule in an in vivo setting, again, showed very good PK profile, very good microsomal stability, no concerns with regard to P450 inhibition, an acceptable aqueous solubility, and mouse PK looked very favorable. And when you took it into an HBV HDI mouse model for HBV, and what you saw here was in fact very nice inhibition of HBV replication in the serum at 3 and 10 milligrams per kilogram. You can see up to a 2.5 log reduction in HBV DNA. And that's when you compare it to the previous generation molecule, AB506, at 100 milligrams per kilogram, certainly a significant improvement. So we saw inhibition both in the serum and actually in the target organ, the liver. So we're very happy with this molecule. In addition, we also show that it solved the resistance concerns that we had with the previous generation agent. And so you can see the I105T, I105V, we saw very nice low nanomolar inhibition of that variant. So looking at PK across multiple species, rat and monkey, we saw, in fact, that we had very good clearance numbers. But one of the things that we were very interested in is a very high liver extraction ratio. And why we were interested in this is simply because Obviously, this is a liver disease. The virus replicates actively in the liver, and so we really wanted to have a molecule that was liver-centric, so to speak. And so this molecule fulfilled that requirement. Preclinical safety profile looks very good. We use this raw reactive oxygen species assay as a new discriminator to see if we can address the liver toxicity associated with 506, the original molecule. That molecule actually showed activity against the Ross ATP assay. This molecule and its metabolites did not. So we felt very comfortable that we've overcome that with this new chemical series. So this molecule was taken into a phase 1 clinical development program in the SAD and MAD portions of the program, and then also in the chronically infected patients. And so what we saw in the SAD, we saw a very nice dose-dependent increase in exposure of the molecule from 10 to 175 milligrams, and again, in multi-dose studies out to 10 days, again, we saw a nice dose-dependent increase in exposure. When we took this molecule in chronically infected patients, again, we saw a very nice dose-dependent reduction in HBV DNA levels down to about 3.5 log drop at a 200 milligram once a day dosing. So we were very, very excited about that. But again, at around day 20, we saw ALT elevation. This was very surprising to us. We had taken this molecule already by this time through chronic toxicology studies in both rat and dog. This molecule showed absolutely clean profile, especially in the liver. And so we were very surprised when we took this molecule then back into healthy volunteers in a 35-day healthy volunteer assessment and saw ALT elevations around day 20, 25 with this molecule. So we were a bit of a loss on where to go with this. So ultimately, we decided to terminate the clinical trial of this agent because of the ALT elevations. And the issue here is clearly, for patients who have a chronic liver disease, any kind of ALT elevation is not good. So right now, this is where we stand with our capsid inhibitor program. The field is now assessing whether these CAMs, capsid assembly modulators, will play a critical role in combination therapy for HBV-cure. So we have a next generation agent that we have waiting in the wings as we begin to assess where this class of molecules, where this mechanism plays a role in ultimately achieving a cure for HBV. Now let me go to, now switch gears to the issue of surface antigen. So, we really want to drive surface antigen levels down very low to be able to achieve this, hopefully, reactivation of the host immune response, knowing that S antigen plays an important role in this immune exhaustion phenotype that you see in HBV patients. So I'm going to talk about two ways to address the S antigen problem. One is active degradation, and the other one is reduced stability. So let's talk about active degradation. So this is an siRNA strategy that we've developed using a GalNAc conjugated siRNA methodology. This GalNAc siRNA is a single trigger, and what that means is a single siRNA, because of the nature of the overlapping reading frames of the HBV genome, one siRNA can take down all viral transcripts. And it's given as a subcutaneous injection and relies on the GalNAc display of the N-acetylgalactosamines to bind to the acylglycoprotein receptor and be internalized into the cell as a delivery mechanism. So the way this works is the GalNAc portions of the molecule get recognized by the acylglycoprotein receptor. It's internalized into the endosomes. You get release of the antisense portion of this. That binds to the risk complex, and then you obviously then get cutting of the viral mRNAs. So this molecule, this siRNA, AB729, is active against all viral genotypes as well as nucleoside-resistant variants. And in an AAV mouse model of HBV infection, you see a very nice dose-dependent decline in HBV S antigen to fairly low levels. And you can see that clearance in electron micrographs of slices from the livers of these animals. One question we asked ourselves was, you see this very nice decline and then this long duration of effect that's happening. What's driving the long duration of effect? Now, what we do know is that once you give these GalNAc conjugated siRNAs, it traffics directly and rapidly to the liver. So, within less than 24 hours, you see no siRNA in the circulating serum. So we did a study to look at this, look at the kinetics. So this is the S antigen reduction over time, administered in an AAV mouse model. And what you see also is that when you look at AB729 antisense strand in the liver itself, you see that it really sort of maximizes its presence around 4 hours and then rapidly declines thereafter. So it turns out that, the duration of effect can't be just ascribed to the concentration of the siRNA in the liver itself. So what we ultimately decided to do was to look at the risk complex itself. And the risk complex is composed of 3 proteins, the dicer, the Argonaute-2, and TRBP, which is a protein which binds double-stranded RNA. And what we saw, if you look at this, again, this is the efficacy plot in the mouse study. And this is the Argo-2 associated complex. And you see what happens is that you get maximal complex formation at around day 14, and then it begins to tail off after that. And that correlates very nicely with the PD effects that you see. So you get maximal effect at day 14, and then the decline over time, which correlates very nicely with what we see as association with the anti. So it's really the risk complex, the stability and longevity of the risk complex that really controls the pharmacodynamics effect that you see in the animal model and ultimately in patients. So this molecule was taken into patients in healthy volunteers phase 1 study. It was very well tolerated and showed robust S antigen decline across all cohorts and HBV DNA declines as well. So what you have to understand, as I mentioned, we cut all transcripts, so we only knocked down S antigen. We knocked down HBV DNA levels. We knocked down core protein. We knocked down what is known as X protein, which was the transcriptional activator for cccDNA. So all proteins get knocked down. So then we decided to look at a dose and duration study, so looking at 60 milligrams and 90 milligrams for either 4, 8, or ultimately 12 weeks, and then look out to 48 weeks and see what's the duration of effect that we see with this siRNA. And what you see now at 48 weeks, you see that in fact all cohorts will be able to reduce S antigen to about 1.9 logs after dosing either every 8 weeks or every 12 weeks. Safety was unremarkable, and there turns out to be no significant differences between the 60 and 90 milligram doses or dose intervals. Now, if you look at the individual patient plots, what you see, so we dosed out to 48 weeks and then observed the patients after that, and you see a very slow and gradual return of S antigen levels, although in most patients, they never get to their original baseline levels. In fact, there are some patients that have actually gone to undetectable and generated S antibodies, but only very infrequently. But we do see is 33 of 41 patients had an S antigen of less than 100 international units. And what this is actually an important number, because in the hepatology field, if you can reduce HBV DNA below 100, that's time when patients who have been on very long-term, 10-year plus therapy of nucleosides, physicians, some physicians at least, have begun to remove nucleoside therapy to see if in fact those patients now retain that undetectable HBV DNA and can progress to functional cure. Will their immune systems ultimately be reactivated? In some very small number of patients, they can. So the next less than 100 international units of S antigen is an important number. The other question we asked was, what's happening now when you reduce S antigen load? Are you getting that reconstitution of the host immune response? And so for patients who consented to having their PBMCs drawn, we actually did an extensive immune profiling, cytokine and immune cell profiling on these patients. And this shows what's happening in a couple of these patients. So you get S antigen decline, you get S antigen, you get ALT elevations, now not very large in some of these patients, but you also begin to see actually immune activation. So these bars represent HBV-specific interferon gamma levels, and you also begin to see sort of a decreased exhausted T cells in these patients. So we're getting a hint that in fact something may be happening as you treat these patients, reducing S antigen load, but it's still not quite enough to actually drive these patients to undetectable. So this molecule now is continuing on in phase 2B clinical development and moving forward. And the question is really, what do you combine it with? So you have a nucleoside, and all these patients were treated with nucleoside plus the siRNA. The nucleoside helps obviously suppress the HBV DNA piece more effectively. So what do you need to add to this? And that I'll address a little later. So HBV, there is another way, and I talked to you about this way as sort of a different way of reducing S antigen. So it turns out from work that we have been done and some others have done, there is actually a mechanism by which the HBV RNA is stabilized. And it turns out that HBV RNA has this PRE region, post-transcriptional regulatory element, that binds two host proteins, or actually three host proteins, but two key ones, PAPD5 and PAPD7, which are non-canonical poly-A RNA polymerase proteins. And they help establish, and this complex maintains the stability of the poly-A tail on the RNA, and also introduces, by introduction of guanylation at various points along this. So through a series of screening efforts, which one was able to show was that identify a molecule, small molecule series that when disrupting this association ultimately leads to degradation of the poly-A tail and therefore degradation of the viral RNA. So one of the molecules that we identified that is able to do this is shown here, AB452, which is known as a dihydroquinazolinone series of molecules. And this was the first generation, what we call RNA destabilizer. It was very potent. In fact, it acted like identical to a siRNA. It was able to knock down all the viral transcripts except X. So you're able to see S antigen reduction in animal models. You were able to see reduction in HBV DNA levels and HBV RNA levels. So it's quite exciting. The reason why it was exciting because this provided a potential for an all-oral combination therapy for an HBV cure. And still today, this is the only mechanism that is able to have that hope. So this molecule was taken into 28-day toxicology studies looked very clean and was actually now progressed and got an approval to go into a healthy volunteer study. Now, at the same time, we were actually doing further toxicology studies, longer-term toxicology studies. A 90-day GLP tox study was underway, and we observed at around day 35, we began to see unusual clinical observations with these animals, meaning they had unusual gait, they had these splayed limbs, and at that point in time, just before we dosed our first patient, first healthy volunteer in the clinic, we put a hold on that study, a clinical study, and began to do some investigational toxicology to understand what was going on. And so we had done a number of studies, and one particular was really to look at nerve conductance velocity in several nerves, the perineal and sural nerve conductance in the periphery, and observed that this molecule caused nerve conductance velocity reduction. It also, in histopathological studies, we also saw some minor spinal cord degeneration. So obviously that's a no-go, although we did see over a fairly long period of time, over 17 weeks after cessation of dosing, we did see recovery to a large extent in these animals. So we ultimately halted the clinical study of this molecule and developed a new strategy to be able, we believe, to be able to move forward here, because we believe this mechanism was really quite unique and provide a whole new paradigm for HBV patients. And that was to actually find a second-generation molecule that was designed to specifically target the liver so that you would get no peripheral exposure. And that molecule is known as AB161. So AB161 is very potent against HBV and reducing S antigen in vitro studies. Is active against all genotypes and all mutant resistance nucleotide analogs. And it was very selective for HBV and had no effect against many, many other viruses that we study. What you can see with this molecule, AB161, this is AB453, the previous generation agent, was in fact that it does reduce core protein, reduce capsid levels. It's very differentiated from a nucleoside if you look at the profile here. And like I said, had no effects against HBV RNA with the capsid structure, where this molecule can actually knock down all HBV RNA levels. And actually, the effect was pretty quick. In fact, within 2 to 4 hours, you began to see the reduction in RNA levels with AB161. So what we also demonstrated was that this molecule actually was an inhibitor of these two enzymes, PAPD5 and PAPD7. And in fact, that the PRE region, the stem loop alpha specifically, was critical. So if you either remove the stem loop alpha or that you reverse the stem loop alpha, what you see is a complete obliteration of activity of the molecule itself. So in an animal model, the molecule showed a nice dose-dependent reduction in HBV RNA. This is again in the AAV mouse model. And as we hoped, we saw preferential liver to plasma exposure. So we're obviously getting very high liver exposures that we were really hoping to get with this molecule. So we got the high liver concentrations and favorable liver to plasma ratio. We saw in the repeat dose toxicology studies, we saw no clinical observations or no liver enzyme elevation biomarkers noted. And in the 60-day non-GLP repeat dose toxicology studies, we were able to demonstrate that we did not observe any of either the clinical observation of splayed legs in these animals or any observation of the nerve conductance velocity changes. So this molecule now is currently in phase 1 clinical development and progressing through the SAD, MAD, and hopefully we'll get into the chronically infected patients. So that takes care of two pieces. So we know we can reduce HBV DNA, we can reduce S antigen levels substantially using several different mechanisms, some now proven clinically in the siRNA case. Next question is, what do we do about immune activation? It's pretty clear that siRNA itself, or the combination of siRNA with a nucleoside, cannot re-engage the host immune response substantially to ultimately lead to this functional cure. So we've been interested in, for a long time, in looking at checkpoint blockade as an approach to address the immune activation component of our strategy. The reason why we've been interested in checkpoint blockade is because we know that PD-L1 expression is really upregulated on hepatocytes in HBV chronically infected patients, and PD-1 is upregulated in HBV-specific T cells and B cells. We have demonstrated, actually clinically, it's been demonstrated by others in a very small clinical trial, that if you give nivolumab, a PD-1 inhibitor, to chronically infected patients at sub-oncology therapeutic levels, that you can get reductions in S antigen, and in one case, a functional cure in a patient. And we've demonstrated in an animal model that a combination of an siRNA and a PD-L1 inhibitor actually ultimately leads to some reconstitution of immune function in those animals. So we were interested, however, in not looking at antibodies, because if you understand HBV patients are fairly well managed on long-term nucleoside therapy. So to introduce an agent that has significant side effects is not something that is acceptable within the field. So you want something that's in fact more tolerable. And so we decided to actually go to a small molecule strategy that we believe that we could minimize systemic exposure. We could actually tune the control of the checkpoint inhibition and better tissue penetration than with an antibody strategy that would have a long-term on effect with an antibody strategy. And so we've identified a series of molecules that bind to PD-L1 molecule, and it turns out they bind as a dimer. So anti-parallel binding of the molecule to two PD-L1 molecules as shown here. And what we've also shown is that they operate by a very different mechanism of action than does a antibody. So in fact, what you see here is either through flow cytometry or confocal microscopy, what we've been able to show that what happens is that you have PD-L1 on the surface, you give the compound, and what happens, you get internals, you get dimerization and then internalization of the PD-L1 protein and ultimate degradation. If you wash out, and that happens within an hour actually, and if you wash out the drug itself, you get reconstitution, new protein synthesis within 48 hours. You get full reconstitution of PD-L1 on the surface of the cell. So we ultimately took this forward in a medicinal chemistry program using structure-based drug design to help guide us to lead optimization and actually improve the intrinsic cell-based activity of these molecules. And so as you see here, it's AB101, which is our lead clinical candidate, small molecule. So it compounds highly potent against cells from chronic hepatitis B patients. So this is percent internalization. So with an IC50 of about 2 nanomolar. And in fact, if you look at, sort of a functional assay where you take PBMCs from chronically infected hepatitis B patients, stimulate them with core HBV peptides, either core or surface protein from core or surface protein, and treat with a PD-L1 inhibitor to look at interferon gamma production. What you see is that AB-101 works as good as nivolumab, as atezolizumab at reactivating these HBV immune cells. Here again, we were very interested in a liver-centric agent. So we developed these molecules that have high liver centricity to, again, circumvent any peripheral exposure for these molecules. And we're able to get very high liver-to-liver concentrations of these molecules in these animal studies. Now, we wanted sort of an efficacy study. At the time, we didn't really have a model that we could go to from an HBV standpoint, so we looked at these molecules in an MC38 tumor mouse model and demonstrated that one can actually, at a 10 milligram per kilogram dose over 28 days, QD dosing, we can actually get equal effectiveness of reducing tumor size as atezolizumab, which is obviously an approved antibody. So this molecule now has completed IND-enabling studies and is currently in phase 1 clinical, going into phase 1 clinical development. So, in summary, we see that chronic viral hepatitis is a major public health issue, which impacts and eclipses many other infectious diseases. So the discovery of oral, short-duration curative therapies in HCV has transformed therapy for those patients and obviously led to cures for over now 5 million people globally. The development of curative therapies for HBV is really focused on a combination therapy that needs to do the three things, really. Reduce viral replication as complete as possible, reduce antigen load, and reactivate the host immune response. We see that AB729 and siRNA can be quite effective at actually reducing S antigen load and beginning to see hints of immune reactivation. We see that a small molecule checkpoint inhibitor has the potential to re-engage or reactivate the host immune response, at least based on preclinical studies. And so the combination of these agents now going forward will be where the field is now going. So you see the field now progressing such that if you look at an siRNA, which is now the most studied agent to S antigen load, now being combined with interferon. Now we're doing a study to combine it with a therapeutic antibody, therapeutic vaccine, as well as our PD-L1 program. So a number of clinical trials in combination that now use a nucleoside to reduce HBV DNA and siRNA to reduce the antigen load, and then trying to figure out what that third component is, which is the immune activation piece. And finally, I'd like to thank all those who were involved in all this work, particularly Andy Cole, who leads the chemistry group and developed all the work on the capsid and PD-L1 efforts. Angela, who leads the chemistry. Troy, who leads our DMPK group and all the efforts there, and our clinical development colleagues who've taken all these molecules into the clinic to look at their potential as therapeutics. And thank you for your attention.
Thank you, Michael. That was fantastic. I would like to open the floor now for questions. I'm sure there are questions. I have a lot of them. So. Then I'll start off. And unfortunately, we only have 4 minutes for questions. So if those of you have some other questions, then please come to see Michael afterwards. So I had a question when I was looking at all this, is that you'd, and truth being said, I'm a pharmacokineticist, so a bit tunnel visioned. Is there a role for studying intracellular kinetics of small molecules? If I look at these diseases, you obviously optimizing the drugs to go into the liver, but within the liver, there's liver cells and different liver cells, and within the cells, there are certain targets. So is that something that we could even account for during the entire drug development process? Do you think so?
So yeah, so we, what's driving that concentration in the liver? There are drugs like sofosbuvir, which are actually designed to take advantage of a certain mechanism to deliver it and trap the molecule in the cell, right? In HCV. For HBV, we've fine-tuned all the characteristics of the molecule to get this liver centricity. Now, what is driving that liver centricity? Some general characteristic of the molecule or some active uptake of the molecule into the liver, we don't know at this point in time.
Right. I have another question. So the small molecule PD-L1 inhibitor is like very interesting, and is it also very likely that it has activity in other diseases, but also that it might actually cause some side effects in that respect?
Well, we've been very interested in not only the PD-L1 small molecule for HBV, but we have a particular interest in hepatocellular carcinoma as a potential utility for these molecules, right? So again, they're highly liver-centric, right? Versus an antibody. So could that be beneficial to cancer patients or HCC patients where they have very few options at this point in time still? So that is a possibility as another therapeutic target area for us. But I think the possibilities are endless really for these small molecules and as checkpoint blockade inhibitors. So.
I have one final question. So you showed repetitively ALT elevations at around weeks 3 to 4 with a lot of different small molecules that had an effect in hepatitis B virus infection. Did you investigate what it actually is that causes that? Or because it almost seems like it might be an effect that you see in every patient, and it may be a sign of the activity of the drug itself?
That's a good question. We really wanted the answer to that because we thought we had the answer when we looked at the second generation molecule 506, and what that molecule we saw, even though it was a small metabolite formed, that we saw in the Ross ATP assay that was pretty active. So we thought it was a metabolite. We believed that we had an assay to address that, to screen for that, and we put our next generation agent through that, and it was absolutely clean, and metabolites were absolutely clean. So we were then really surprised in the end when we saw this, especially when we had long-term toxicology studies that showed there were no liver effects at all with this the third generation agent. We don't know. We just don't know what it is, and that's. We would love to figure that out, but at this point in time, it's going on to other things.
Okay, thank you. If there are no further questions, then I'd like to thank you very much for a very inspiring talk. Thank you.
Good afternoon, everybody. I'd like to welcome you to this oral communication session on cardiovascular. My name is Cherry Wainwright. I'm from Robert Gordon University in Aberdeen, and my pleasure to be Vice President for Meetings for the British Pharmacological Society. And this is my co-chair.
Thank you for the opportunity. I'm Monica Mukhia, and recent graduate from University of Glasgow, Masters in Clinical Pharmacology. Thank you so much.
Okay, thank you. So our first speaker this afternoon is Paraskevas Sampas from the Biomedical Research Foundation of the Academy of Athens, who is going to speak to us on hydrogen sulfide donors' improved cardiometabolic profile in a mouse model of metabolic syndrome.
Thank you very much. I'm actually substituting for my student who couldn't make it to Glasgow. I'm the senior author, I'm Andreas Papapetropoulos, and I'll be talking to you about hydrogen sulfide donors in cardiometabolic syndrome. The metabolic syndrome is a clustering of medical conditions that are associated with problems in the cardiovascular system or in metabolism. So to be classified, for humans to be classified as having metabolic syndrome, they need to have a blood pressure of over 130, or fasting blood sugar over 100, a waist larger than 40 inches, and abnormal values for HDL, abnormally low values for HDL, and abnormally high levels of triglycerides. There's a growing number of people throughout the world with metabolic syndrome, and to introduce our molecule of interest and our pathway of interest, which is hydrogen sulfide. The hydrogen sulfide pathway consists of a number of enzymatic reactions that will give rise to hydrogen sulfide, and a number which are depicted on the left-hand side, and a number of enzymes and pathways that will degrade hydrogen sulfide. On the left-hand side, you can see enzymes that the transsulfuration pathway, CSE and CBS. What is my pointer? This is my pointer, right? Okay. The CSE and CBS that are part of the transsulfuration pathway using homocysteine or cysteine to generate hydrogen sulfide. There's a third enzyme in the pathway, 3-mercaptopyruvate sulfotransferase, that uses a breakdown product of cysteine, 3-mercaptopyruvate, to yield hydrogen sulfide. There's additional sources of hydrogen sulfide in our bodies. H2S can be made through gut microbiota and also through non-enzymatic reactions, and there's also the H2S that we consume. On the right-hand side, you can see the pathways that are responsible for breaking down hydrogen sulfide, and the main enzymes here are sulfide quinone oxyreductase, abbreviated as SQR, ethyl-1 rhodanase, also known as TST1, and sulfide oxidase. So hydrogen sulfide has been linked to a number of conditions that are associated with the metabolic syndrome: obesity, hypertension, type 2 diabetes. Hydrogen sulfide is usually linked in a therapeutic way with these pathways, meaning if you provide hydrogen sulfide, you are able to ameliorate obesity, reduce hypertension, and improve the complications of type 2 diabetes. Our own group has done quite a bit of work on hydrogen sulfide and obesity. We find reduced levels of hydrogen sulfide in obesity, and especially the enzyme MPST, or 3-mercaptopyruvate sulfotransferase, is responsible for restricting excessive weight gain in animals fed a high-fat diet. So with this in mind, we asked two questions. We asked whether endogenous levels of H2S are altered in the metabolic syndrome in a mouse model, and whether we can, if they're altered, whether we can correct the parameters of metabolic syndrome using pharmacological approaches. A few words about our model. We use a mouse model where we feed mice a high-fat diet, 40% of calories from fat for 10 weeks. Then we introduce L-NAME to inhibit endothelial nitric oxide synthase for an additional 5 weeks, and then look at the end of the 15 weeks what happens to their body weight, blood pressure, and hyperglycemia. This looks very much like the two-heat model that is used in the HFpEF literature described by Joe Hill's group. Only these mice in the heart failure model are fed a much higher high-fat diet with 60% of the calories coming from fat and L-NAME throughout the duration. This model that I'm just describing does not lead to the development of heart failure with preserved ejection fraction. So a characterization of our model in this first slide: mice receiving a controlled diet don't gain as much weight. The mice that receive a high-fat diet gain more weight. L-NAME doesn't really change that. And in terms of blood glucose levels, you see that obesity leads to impaired glucose handling and increased and glucose tolerance, and this is obvious in the mice with a metabolic syndrome. Obviously, due to the L-name administration, we also have increases in systolic and diastolic blood pressure in the groups of mice receiving L-name. And surprisingly, only the group that has metabolic syndrome has an increase in the heart weight to tibial length, suggesting cardiac hypertrophy, and increased in mass in the different adipose tissue depots, inguinal and gonadal white adipose tissue. So we do have a model that's working, and it recapitulates the features of metabolic syndrome in humans, and we went ahead and characterized the levels of hydrogen sulfide and the different enzymes in the aorta and in the heart. So looking at the H2S synthesis enzymes, we observed that CBS and MPST are reduced in the aorta of mice with metabolic syndrome, and also the enzyme that's responsible for breakdown, TST, is also reduced in these mice. Unlike what we observed in the aorta, the levels of the enzymes for both the synthesis and the degradation of H2S were not altered in the heart. Given that we found reductions in both H2S synthesizing enzymes and H2S degrading enzymes in the aorta, we went ahead and measured actually H2S in the tissues, and there's two different pools. There's free sulfide and there's sulfane sulfur. Both of these pools were reduced in mice with metabolic syndrome, in line with our observations that no changes in the enzyme levels of H2S synthesizing or degrading enzymes occurs in the heart. We observed no change in the levels of H2S in the heart. So we have established that the levels of H2S in metabolic syndrome go down in this mouse model, and we wanted to see if administering H2S donors would correct the parameters of the metabolic syndrome. So in this slide, we introduced the treatments. We do the 10 weeks of high-fat diet, and then on week 10, we introduce L-name to increase blood pressure, and at the same time, we give either GYY 4137 in IP or SG1002 in the food. Both of them are hydrogen sulfide donors, and GYY is a hydrogen sulfide donor, SG is more of a polysulfide donor. What we observed is that the mice receiving the sulfide donors have reduced body weight, and they have somewhat improved glucose handling as judged by the glucose tolerance test. We have also observed that the mice receiving GYY have reduced systolic and diastolic pressure. There's a trend for the SG group to decline, but this did not reach significance. So also in the heart, the hydrogen sulfide donors, especially GYY, can reduce the heart to weight, and this treatment with the sulfide donors did not alter the mass of the adipose tissue depots that we studied, inguinal fat and gonadal fat. In the heart, GYY was able to reduce cardiac fibrosis, as depicted on this slide, during metabolic syndrome, and the increase in blood pressure. This leads to fibrosis in the heart, and we were able to reverse that with GYY. There's also, again, a trend here with the SG compound, but this does not reach significance. To conclude, metabolic syndrome causes changes in hydrogen sulfide metabolism, resulting in a reduction of endogenous H2S levels in the vasculature, and pharmacological interventions based on H2S restoration could be effective in metabolic syndrome treatment. I'd like to end by thanking our collaborators in this study, the funding sources, and this is a very important slide for me. We'd like to invite you to the EFAR conference 2024 in Athens next year. Thank you for your attention.
Can you switch my microphone on, please? Thank you. I see somebody is already at microphone number 2. If you would like to ask a question, if you could make your way to one of the standing mics, and I will keep an eye open. So, yes.
Rebecca Ritchie, Monash. Thank you for a nice, clear presentation. Have you looked at the effects of H2S metabolism in smaller vessels, the coronary microvasculature, resistance vessels?
We'd love to, but we have not. I know that the relevance would be in the smaller vessels, and some of our ongoing studies to study contractility and how that is impaired and endothelial dysfunction are unfortunately in the aorta. We'd love to collaborate, though.
Thanks. And a very quick follow-up question. Have you looked at perivascular fibrosis?
We are looking at that, but these studies have not been completed. That's a very good idea. Thank you.
Okay, one here at 2 and then at 3.
Thank you. I'm from Mexico City, and I wonder if you have tried to look for reactive species, oxygen species, for this effect of hydrogen sulfide.
This makes perfect sense. Your suggestion is in line with what we would expect in metabolic syndrome, increased levels. We have not measured them. We would expect they are increased, and we would expect that supplementation with donors, hydrogen sulfide donors, would actually ameliorate that, but we have not measured it. We've measured it in other contexts, in different types of experiments, in obesity or in heart failure, but we've not done it in this particular model.
Yes, thank you.
Thank you very much, Andreas, for a nice presentation. This compound, this H2S donor, by itself, does it change blood pressure or heart rate?
The short answer is it depends on which of the two you are, SG or the GYY?
Yes, both.
Okay, but the GYY will reduce blood pressure, and SG will cause a decline that in our model is not statistically significant. Throughout the literature, at this dose, which is probably the maximum dose that has been used, doesn't do too much in the blood pressure. To get a reduction in blood pressure, you need heroic concentrations, and the entire field with H2S and hydrogen sulfide, as you probably know, in terms of regulation of blood pressure, has lower concentrations doing one thing and higher concentrations doing another.
Can I just ask a question? I think, did you say at the beginning that this model, you don't see the functional changes that you would expect with HFpEF?
Yes, that's correct.
But later on, you showed that there's quite clear hypertrophy and cardiac fibrosis, which I would have imagined would result in some cardiac dysfunction.
Thanks for pointing this out. When we do the echo measurements and we look at ejection fraction, there is no difference. And when we do the E to A prime and E to A ratios, this is not different either. There is this hypertrophy. So you don't have the classic, we haven't measured the pressures.
So you've not done like pressure volume loop or anything?
Yeah, we haven't done all of this, okay. but echocardiographically, it seems that this model does not drive the animals into heart failure. Now, if you're taking away enzymes like use 3-MST knockout mice, you'll drive them into heart failure. So I would say it's a borderline, the best I can describe.
Okay. Thank you very much.
Thank you.
Thank you so much. Going onward to our session, we would like to welcome Mohammad Adim Abdul Ghani on carboxyl amylorides, the cardiac fibrosis and the hypertrophy in doxorubicin-induced cardiotoxicity rat model.
I'm just getting a signal that I don't think that speaker is here. Is that correct? Yeah. Okay. I'm just conscious of the fact that we are supposed to be staying on time, parallel with the other sessions, so people can move backwards and forwards. So I think my best option is to take a short break to allow people to move between sessions so that we keep to time. So apologies for this, but we will reconvene in just over 10 minutes. Thank you. Apologies, there seems to have been some confusion or the slides that the speaker put in did not appear to have been uploaded properly. So what I'm going to do is I'm going to push that communication back to the end of the session. I know it will go into the coffee break, but at least we'll keep on time for the other presentations. So please bear with us and apologies. As they say, the best laid plans of mice and men, which is a good phrase, I suppose, for a pharmacology meeting. There apparently had been a last-minute change of a speaker presenting the previous abstract, so we are hoping to have that included at the end of this session. We know it will run into the coffee break, but at least we can make sure that that person gets the opportunity to present. So I'm going to hand over to my co-chair now to introduce the next speaker.
So going forward, let's welcome Baofeng Yang from Harbin Medical University on topic on new targets for cardiac ischemia for fusion injury, INCRNA. Let's welcome Baofeng Yang.
Okay, thank you.
She will be here in a couple of minutes.
No, I'm sorry. We need to stick to the time for the actual speaker, so we need the speaker here now. Thank you. Do we have our speaker yet? Okay, I'm sorry, but I think we're going to just have to move on to the next speaker, and we'll wait, see if this other one turns up. So apologies for the mistiming then, but the next speaker is, who I hope is here, is Jose Brito Jr. from the State University of Campinas to speak on 6-nitrodopamine is the most potent endogenous positive inotropic agent in the rat-isolated heart. Thank you very much.
First of all, I would like to thank organizers the opportunity to present the work you have been developing on 6-nit dopamine. For the last couple years, you have been investigating the release and action of 6-nit dopamine in various organs. For instance, 6-nit dopamine is released by human umbilical arteries and they are detected by liquid chromatography compared to tender mass experimentally, LC mass. In the human umbilical cord vessels, 6-nit dopamine acts as a potent vasodilator, antagonizes the contractile response of dopamine at the Q-like receptors. In head-isolated right atria, present basal release of 6-nit dopamine, whereas 6-nit dopamine acts as a potent positive chronotropic agent in these some cases. Actually, 6-nit dopamine is 100 more times potent than noradrenaline and adrenaline as a positive chronotropic effect, and 10,000 more potent than dopamine as a positive chronotropic agent. Another interesting characteristic of 6-nit dopamine in the head isolated right atria is that 6-nit dopamine presents a remarkable ability to synergize with noradrenaline, adrenaline, and dopamine in the positive chronotropic effect induced by the classical catecholamines. As measured by LC mass, 6-nit dopamine is released by rat isolated ventricles, and as described before, the release of 6-nit dopamine is reduced when the ventricles pre-incubate with the nitric oxide synthase inhibitor, L-name, and when the ventricles were obtained from animal chronically treated with L-name. It is important to indicate that incubation with L-name did not a significant reduction in the basal release. 6-nit dopamine, basal release 6-nit dopamine, but it did not abolish, indicate that the other biosynthetic pathway for 6-nit dopamine. Interesting, I showed this slide, incubation the ventricle with the sodium channel blocker, tetrodotoxin, did not affect the basal release of 6-nit dopamine, indicate that the release of 6-nit dopamine is unlikely to be from neurogenic source. Rat isolated heart Langendorf, heparin as injected to prevent blood control, and anesthesia was performed by isoflurane overdose. The isolated heart was performed with chloride solution, equilibrated with carbogen gas mixture, and constant flow 10 ml per minute, and the pressure 80 to 10. As to recollect the Langendorf preparation, here is a short illustration of the said procedure. This is a typical trace obtained from the isolated hearts. Here, it can be observed the increase in left ventricular developed pressure induced by a single bolus of 6-nit dopamine at the dose 1 picomole, and the increase in left ventricular developed pressure induced by a single bolus of noradrenaline, adrenaline, and dopamine at a dose of 1 nanomole. Yes, 6-nit-dopamine is 1,000 times more effective than other classical catecholamines. Here, using the het perfusion heart model, a single bolus eject of 10 femtomole of 6-nit-dopamine causes a significant increase in the left ventricular developed pressure. At this dose, 6-nit-dopamine is 1,000 times more potent than the classical catecholamines, noradrenaline and adrenaline, and 10,000 times more potent than dopamine as a positive inotropic agent. Again, observing with the inotropic effect 6-nit-dopamine is the het isolate heart rate, the effect is apparently not dose dependent. Here are the results obtained with the maximal rate of Foresite of the left ventricular developed pressure, the dp/dt max, on more the left ventricular developed pressure itself, a single bolus inject of 10 femtomoles of 6-nit-dopamine causes a significant increase in the dp/dt max. Furthermore, here are the results with the rate pressure product, defined as the product of a heart rate and left ventricular developed pressure, as seen before with the left ventricular developed pressure itself. A single bolus inject of 10 femtomole of 6-nit-dopamine causes a significant increase in the pressure product. As noted the introduced slide, 6-nit-dopamine has a potent inotropic positive action in the het isolate atria, and it has a remarkable ability to synergize with noradrenaline, adrenaline, and dopamine. For this slide, we have infusion for 1 minute 6-nit-dopamine at the final concentration of 0.1 femtomole. At this concentration, 6-nit-dopamine does not present positive inotropic action in the het isolated heart. However, a single bolus of 0.1 picomole of noradrenaline causes substantial increase in the left ventricular developed pressure. As expected in the slide, the minimum dose of noradrenaline to increase left ventricular developed pressure of 10 picomole. This remarkable potent of the inotropic effect of noradrenaline induced by 6-nit-dopamine, as was observed with adrenaline and dopamine. In conclusion, the result demonstrates that 6-nit-dopamine, besides being the most potent endogenous positive inotropic agent so far described, has a remarkable ability to synergize with the classical catecholamines. 6-nit-dopamine presents a novel therapeutic approach for the treat whether acute or chronic heart failures.
Thank you very much. Do we have any questions from the audience? Yes, mic 3. Microphone 3.
Ulf Simonsen from Denmark. I was just wondering whether you can block the effect with a beta blocker.
The beta block?
Yeah.
The beta block abolishes the increase inotropic action of sedative dopamine, but is more important than the sedative dopamine, either adrenaline and the dopamine. For example, atenolol 10 nanomole don't block adrenaline, noradrenaline, and dopamine action inotropic in the Langendorf, but abolish the effect of 6-nit dopamine.
I'm not sure I understand you, but if you take propranolol, for instance, and infuse that in your preparation, can you then block this 6-nitrodopamine response?
Propranolol, I don't use propranolol here, but so atenolol.
Okay, fine, thank you.
Thank you.
We have in mic 2, yes.
Rebecca Ritchie, Monash University. What can you tell us about the vehicle you used, and did that impact pH of the buffer? And what are the pharmacokinetics of 6-nitrodopamine?
Pharmacokinetics. Repeat me.
Okay. What did you dissolve 6-nitrodopamine in?
What is desserts?
What did you make it up in?
Sedative dopamine is released from umbilical.
Yeah, when you were adding it to, you said you added the 6-nitrodopamine.
Water. It's diluted in water.
Thank you, thank you, thank you.
Water.
Thank you. I have a challenging accent. If you're proposing to use 6-nitrodopamine for chronic heart failure, what are its pharmacokinetics in vivo? How long does it last? Is it broken down? Is it cleared?
Well, we haven't been able to measure 6-nitrodopamine in blood levels so far because it's so potent, so the concentration must be very, very low, okay? So we haven't been able to detect circulating levels of nitrodopamine. So at the moment, we have commissioned to make a label nitrodopamine in order to be able to study the kinetic of 6-nitrodopamine half-life in vivo. However, the effect is very long-lasting.
Thank you.
Thank you.
Okay, thank you very much. I think a little bit of confusion has been cleared up, and in fact we do just have one more speaker this afternoon. The topic is new targets for cardiac ischemia reperfusion injury. And can I invite Honglin Zhu to the stage to give the presentation?
Hello, everyone. So today's speaker should be the Professor Yang, but because of some personal reason, he can't come. So on behalf of him, I will give you a talk. So today I'm going to talk about the role of linked RNA-mediated epigenetic regulation in the heart injury. Cardiovascular disease remains the leading cause of deaths in the world. The numbers of deaths owing to cardiovascular disease is about 80 million in 2020 all over the world, and estimated to be 23.6 million in 2030. Statistic data reveal the prevalence of cardiovascular converts from the rising burden of cardiovascular disease, ischemic heart disease, and ischemic stroke, declining the incidence and the mortality of hemorrhagic stroke. To develop effectively and timely strategy to cope with the challenge of cardiovascular epidemics, since 2010, more than 30 drugs for the treatment of cardiovascular disease have been approved by the FDA. Even so, the identification of novel molecular targets and the development of effective drugs for the treatment of cardiovascular disease remains the current top priorities. 95% of the genome is non-coding RNA, which also indicates the importance of non-coding RNA in the body. Non-coding RNA include structural RNA and regulatory RNAs. Beyond their applications as diagnostic and prognostic biomarkers, non-coding RNA can also be the targets of tools for novel therapeutic strategies. Non-coding RNA have been identified as critical novel regulators of cardiovascular risk factors and cell functions and are emerging as fundamentally novel therapeutics. Researchers have developed complex tools and modulated these non-coding RNA with the aim of establishing novel next-generation strategy to combat cardiovascular disease. Another important non-coding RNA is lincRNA, which also been the focus of research in recent years. LincRNA are a class of non-coding RNA molecules of longer than 200 nucleotides present in the nucleus of cytoplasma. More than 40,000 lincRNAs have been found in human tissues, and according to their position in the genome, lincRNA can be categorized as sense, antisense, bidirectional, intronic, and intergenic. Unlike protein-coding genes and microRNAs, lincRNA are often poorly conserved between species and are highly tissue-specific. LincRNA research started later than microRNA research, and after more in-depth research, lincRNA has also the potential to become a drug therapeutic target. The growing evidence had demonstrated that the lincRNA ZFAS1 and antisense lincRNA to the 5' of the protein-coding gene ZNFX1 had significant role in the regulation of tumor. In our previous study, we identified ZFAS1 as an independent predictor of acute MI, and additionally, we found that ZFAS1 bound to and inhibited the intracellular level and activity of sarcoplasmic reticulum, calcium ATPase 2A, and contributed to the impairment of cardiac contractile function in MI. And last year, we published the paper that lincRNA ZFAS1 promote myocardial ischemic reperfusion injury through DNA methylation-mediated Notch1 downregulation. Therefore, ZFAS1 might be considered as a new therapeutic target for preserving cardiac function. The calcium homeostasis, a critical determinant of cardiac contractile function, is critically regulated by SERCA2A. Our study found that ZFAS1 was rapidly increased in both ISR and the cytoplasma of MI heart, and such an upregulation produced a significant negative effect in MI heart. And at the molecular level, ZFAS1 was found to directly bind to SERCA2A protein on one hand and to repress expression on the other hand. Moreover, the NF-ATC2 account at least partially for the upregulation of ZFAS1 in MI. And our papers have several citations in Circulation Research and Cell Calcium. The myocardial ischemia reperfusion injury has been correlated with oxidative stress, autophagy, ER stress, and epigenetic alterations. Although there are many treatment strategies, the incidence remains high, which indicates it's very important to find new targets. So next is the experimental timeline of the MI-RI model. First, the left anterior descending coronary artery was ligated for 30 minutes to cause myocardial ischemic. Then the line was released for 48 hours to allow for reperfusion. And this is the result part. From the left panel, the echo result, we can see in the MI-RI mice, the cardiac function was reduced, and ZFAS1 expression was dramatically elevated. And in cardiomyocyte exposed to hypoxia reoxygenation, the cell viability was reduced, and ZFAS1 expression was elevated. And next, we inhibited the ZFAS1 with AAV vectors to examine the ZFAS1 function. So we could see the ZFAS1 knockdown significantly improved miRNA-induced impairment of cardiac function. And furthermore, SHZFAS1 reduced the infect size of miRNA heart. And next, in vitro, we transfected SIZFAS1, and we could see the SIZFAS1 could mitigate the hypoxia reoxygenation-induced reduction of cell viability and cytotoxicity, showed the similar result. The next is the tunnel staining result, which indicate that the apoptosis was restored by SIZFAS1, and the enhanced ROS generation induced by hypoxia reoxygenation was also reduced by SIZFAS1. Next, to understand the molecular processes supporting miRNA by ZFAS1, we constructed cardiac-specific ZFAS1 knock-in TG mice, and we can see that ZFAS1 was upregulated. Then we compared the transcriptomes between TG and the wild-type heart by gene expression profiles. The experiment revealed that 30 downregulated genes and 90 upregulated genes in TG mice. And GO functional analysis also of these 49 differentiate expressed genes was then conducted with the cluster profile bioinformatic tools. So next, we predicted and screened the direct target of ZFAS1 from 4 pathways, including activation of cardiovascular physiology, stress response, programmed cell death, and cardiac development and apoptosis process. And we identified that the Notch1 and the NTRK3 might be the potential targets of ZFAS1. Because Notch signaling pathway is one of the top 30 rich KGG pathway according to a KGG pathway enrichment analysis of differentially expressed. Therefore, our follow-up research mainly discussed the role of Notch1 in miRNA. The Notch1 expression was identified in TG mice to examine the influence of ZFAS1 on Notch1, and Notch1 mRNA levels were significantly lower in TG mice, which is negatively connected to ZFAS1 expression. And similarly, the Notch intracellular domain in TG mice was considerably lower than in the wild-type mice, which was verified by the immunofluorescence. The Notch signaling pathway is involved in a variety of heart functions and is composed of 4 Notch proteins and 5 ligands. The Notch protein is cleaved into the Notch intracellular domain and released into cytoplasm when the ligand on the neighboring cell surface interacts with the Notch receptor. And after being cleaved, the Notch intracellular domain enters the nucleus and binds with the DNA binding protein, which triggers downstream gene transcription, such as Cas1 and HEY. Inhibiting the Notch signaling has been shown to alter the gene supply of cardiomyocytes, leading to cardiac dysfunction. And from the result, we could see in TG mice, the mRNA and protein level of Cas1 and HEY1 downstream regulators of Notch signaling pathway was both reduced and compared with the wild-type mice. And next, we characterized the regulatory effect of ZFAS1 on the Notch signaling pathway in miRNA and found that the mRNA level of Notch1 was reduced during hypoxia reoxygenation, which was reversed by ZFAS1 transfection. And this result showed that inhibiting the expression of ZFAS1 remarkably increased NICD protein levels. And also in vitro, we used neonatal mouse cardiac myocytes, which exposed to hypoxia reoxygenation damage, and similar expression modifications of HEY1 and HES1 were consistently found among the constructs. So how does lincRNA ZFAS1 regulate Notch1? NOTCH1 is because ZFAS1 is localized within the nucleus of cardiomyocytes. These facts promote us to conjecture that ZFAS1 might regulate NOTCH1 transcription through an epigenetic mechanism, such as DNA methylation. We found a substantial CpG element in the promoter region of NOTCH1 using the UCSC genome browser and mass primer to predict the probable domain of NOTCH1 for DNA methylation. And the methylation inhibitor dramatically enhanced the transcription level of NOTCH1 in cardiomyocytes. And next, in TG mice, the bisulfite sequencing revealed a large increase in methylation at the NOTCH1 promoter region. So how does link RNA ZFAS1 promote DNA methylation of NOTCH1? Link RNA can limitate the gene transcription by binding to DNA and build DNA RNA triple, which can accrue DNA methyltransferase and increase DNA methylation modification in target areas. So we use the RNA tools to see its expression. We can also see the direct physical interaction between ZFAS1 and NOTCH1 promoter region was also verified by means of chereptide with ZFAS1 antisense as a negative control. And the IP result also shows that the DNMT3B bind to NOTCH1 promoter CpG element was considerable, and according to these findings, ZFAS1 bind to NOTCH1 promoter areas and recruit the DNMT3B to enhance NOTCH1 DNA methylation. Then we used the nucleotide replacement, and we engineered a mutation in the ZFAS1 sequence to disrupt the NOTCH1 binding site and investigate the impact of ZFAS1. However, the cell results show that when the cell transfected with mute ZFAS1, the carried plasmid lost its function, and we could see the capacity of mute ZFAS1 to modify the cell viability, cell apoptosis, and ROS levels was likewise lost. These results indicate that the binding of ZFAS1 to the NOTCH1 promoter region is a critical step in its impact on mRNA. And the right part is the drug discovery part, and because of time limitation, if any of you interested in this part, you can see our papers, which published on GACC. So in summary, upregulated ZFAS1 facilitated the DNMT-NOTCH1 interaction and promote DNA methylation-mediated NOTCH1 downregulation. And according to our current findings, which explains how upregulated ZFAS1 underpins the gene genesis of mRNA, and by interfering this epigenetic process, the NMN could used to treat mRNA as a potential therapeutic therapies. Okay, thank you for your attention. Yeah.
Considering the time, we can go for one quick question.
Yeah, because of the time, I need to catch my train, so I need to leave.
We have time for one question.
Okay.
Okay, I don't see any questions out there. We have actually overrun our time now, so I would just like to thank you and the remaining, all the other speakers this afternoon for their presentations, and again, apologies for the disruption to the program. Thank you very much.
Thank you so much.